Great.
Excellent. So moving on to the last session of the day. So this is kind of building on a theme from last year around standardization and breath analysis, and how to start to understand what is on the breath of heterogeneous control populations, normal populations, and then leading into disease cohorts. So building out a BBC atlas. So we've got sessions tomorrow as well. Really excited to welcome partners from the FDA who are giving 3 talks. And what we'll do is we'll take questions in the Q&A as we go through. But all the speakers will come on at the end, and we'll discuss all those questions in a panel type discussion. So really excited to have Victoria, Eric, and Omar talking about the wonderful work that the FDA are doing around standardization and understanding what is on breath. Fantastic. That's working perfect, Eric. Thanks. Over to you.
Great. Good afternoon, and thank you very much, Billy, for the introduction and the opportunity to present our work. I'm Eric, and I would like to introduce and thank our team for all of their fine work. Samantha, Jamie, Krishna, Chatarika, Mark, and Victoria. Today, we will be presenting on confident identification of breath matrix compounds and RST to serve the breath community. First, I want to mention that this presentation reflects the team's views and not those of the FDA, and we do not endorse any of the products mentioned. Our project aims to develop a regulatory science tool to facilitate confident breath compound identification. Breath is highly complex and variable within an individual, as well as across sample populations, with over 1,000 VOCs having been identified with varying levels of confidence. It's not feasible for a biomarker discovery lab to confirm the idea of all these compounds by running certified reference standards due to price or the fact that they simply aren't available commercially. Therefore, identification tools are needed to perform a non-targeted analysis and confidently identify these compounds, such as spectral library match, retention index, MS fragmentation, isotopic patterns, and high-resolution accurate mass instruments. Currently, there is ambiguity in the criteria underlying these tools, resulting in a lack of consensus even on the definition of endogenous versus exogenous breath compounds. This regulatory science tool intends to provide a benchmark through a public breath database that can offer confidence in breath matrix and biomarker identification. Our current areas of focus consist of developing an identification workflow and criteria, collecting reference standard spectra and gas chromatographic data, curating a breath library from standards which were shown in the literature to be found on breath, and generating delta retention index criteria, or delta RI. In layman's terms, delta RI is a difference between an analyte's normalized experimental retention time and that of a reference standard. The smaller the delta RI, the greater confidence that these are the same compound. The highest level of identification confidence in chemical analysis is when performing a targeted analysis in which the certified reference standard is purchased and analyzed by the same lab. The identity is confirmed if the mass spectrum and retention index match within a very low tolerance. However, when the reference standard is not available in-house, commonly referred to as a non-targeted analysis, the level of identification and the process of identification gets a bit foggy, as represented by the gray area in this figure. There are many pieces of information that we, as analytical chemists, use to corroborate the identity in lieu of a reference standard. Many of these pieces of evidence are listed here in no particular order on the vertical axis. What we intend to do is streamline the identification process and prioritize these criteria so that there is interlaboratory consistency. So when we refer to confident ID, we are referring to this gray area above unknown or partially known and below confirmed. This gray area may consist of sublevels such as confident, corroborated, tentative, or speculative, just as examples. Eventually, these pieces of evidence will fall into an organized identification workflow with defined confidence levels. Here is a flow chart of our project showing our current low resolution single quad on the top and the high resolution thermal desorption GC mass spec, which we expect to arrive next month, on the bottom. Once we have it up and running, we will run all the reference standards on high res, which we previously ran on single quad. We will also collect breath samples from healthy subjects as a way to test our confident ID criteria, data processing, and workflow. Here is the current list of reference standards of which we have collected mass spectra and retention indices. These were chosen and purchased based on breath compounds cited in the literature, including Alstone's very recent publication in Metabolomics. We prioritize compounds with the highest statistical significance and frequency of occurrence across subjects. The Drabinska reference shown here is a review of breath research citing specific studies relating to over 1,000 breath compounds. So if you're interested in the exact source, I recommend looking through this review, particularly the supplementary info. Now I'd like to turn it over to Chatarika and Mark, the two ORISE fellows that have worked so hard to collect this data. Chatarika will begin by showing us some of the challenges and limitations of using mass spectral libraries.
Hello everyone, I am Chaturika Ratnayaka and now I am going to show our current progress of the results. Thank you, Eric, for the introduction. We'll briefly discuss the current results. So to ensure the confident chemical identification, a crucial first step after updating the mass spectrum is mass spectral library matching. So for that, we should focus on two main aspects. The first one is the comprehensive and relevance of our library. And the second one, the criteria for evaluating how closely an analytical spectrum align with the reference spectrum. Let me share an example for our preliminary data. You can see over the figures over here. We investigated the impact of the library size on the match factor by selecting a reference standard spectrum of heptane and search the NIST-23 library containing 500,000 of compounds. So on your left side, you can see our search for the heptanals. And not surprisingly, heptanal was an excellent match with a forward and reverse match factor of over 900 and a match probability of 61%. But there are several with good matches of over 100. For example, 4-methylcyclohexanal showed decent head-to-tail match factor of 807. The spectrum is showing the top-bottom over here. With such a large library and so many compounds, specifically hydrocarbons, with identical masses, it's no wonder we see confident library matches. So, next. Okay, well, what if we limited our library search to only those compounds reported by the literature to be on breath specifically? On the left is the search against the full NIST library, and on the right is search for against library of just over 1,000 compounds from the literature. You can see over here, match of the heptanal wouldn't change. It's around 904, but the other compounds probability and match were reduced compared to the large library. Eliminating irrelevant compounds significantly boosts both the likelihood and confidence of this library match compared to other compounds present in the breath samples. So from that, I will turn into Mark, who will show some results of our chromatographic criteria and the retention index. Thank you.
All right. Hi, everyone. I'm Mark Annunziato, and I'd like by first discussing our work with retention indices. To achieve the highest confidence in chemical identifications, it is essential to gather as much corroborative evidence as possible. In GCMS analyses, the use of retention indices to support chemical identification has become increasingly popular. However, the lack of uniform criteria for defining a good RI match poses a challenge, potentially leading to improper application and hindering the effectiveness of this tool in identification efforts. To implement retention index corroboration effectively, we need to establish clear acceptance criteria for both good and poor matches. This involves appropriately populating a database with good RI data and creating well-defined benchmarks for retention indices. To this end, we have begun investigating how our experimental RI values for relevant breath compounds compared to those reported in NIST. Using an HP5 column, which is a 5% phenyl polydimethylsiloxane column, which is equivalent to NIST's semi-standard non-polar values that they report. Our ongoing work aims to define what constitutes a good RI match, establishing a non-exclusionary range and identify thresholds for non-matches in compound searches. While the RI system is very beneficial for identifying volatile compounds, there are certain drawbacks such as the lack of reported data for non-standard columns, such as the midpolar 624 GC stationary phase, which comprises a 6% cyanopropylphenyl and 94% polydimethylsiloxane, despite its popularity or growing popularity in bioanalysis and metabolomics. This lack of reference RI data for compounds analyzed on the 624 stationary phase limits our ability to identify unknowns effectively. So in our study, we started by measuring breath-relevant compounds using direct injection and headspace techniques using both a trace gold 624 column and a DB 624 column. Our goal is to compare the experimental obtained RI values with the closest semi-standard non-polar values reported by NIST. As we anticipated, there is significant difference between the midpolar characteristics of the 624 column and the large delta RI values compared to NIST. To address this gap, we are actively collecting RI values for these relevant breath compounds to develop a more comprehensive retention index library. Additionally, we are reviewing the literature for reported RI values for these breath matrix relevant compounds analyzed on 624 columns to assess variations and establish an appropriate range for delta RI. Now I'll turn it back over to Eric so he could conclude our portion of the presentation.
Thank you, Mark and Chatarika, for your wonderful work that you've accomplished here at the FDA. This concludes our presentation. So before I turn it over to Omar, we'd like to acknowledge the following groups. Alstone for inviting us to present at this conference, the Bill and Melinda Gates Foundation, NIST, the FDA for supporting our work, and ORISE for providing very talented scientists to run the experiments, process data, and present results. Thank you all for your attention, and now I'd like to turn it over to Dr. Omar Rivera.
Wonderful. Thank you so much for that, Eric. And a quick reminder to everyone, post the questions in the Q&A on the right-hand side. So first question here is probably more for Victoria. I think a lot of people that are more involved just on the pure research side of things. I was wondering if you could explain further in layman's terms, what are regulatory sciences tools and why are they important? for those
Sure.
different types of users?
Yeah, so that's why I tried very hard in my presentation to cover it, but if you haven't used it before, it could be very difficult in first pass to understand what they are. Regulatory science tools could be, they are simply a, in our area, let's pick an example. In analytical chemistry, they can be a recipe for how to do an experiment and how to analyze it. It could be an analytical method, which would include sample preparation, the analytical method, and how would you set up your system, what would be your parameters, and it would guide you in a way how to do an experiment. It becomes very important when you are looking at an area where maybe the ISO is not covering it. Maybe there is a gap in the standard and you need some understanding how to do a certain experimentation. Therefore, the RSTs are aimed to address those gaps and teach you how to do the experiment and get the data in support of your submission. That's the aim that we have with the RSTs. Ultimately, what we would like to do is to either help close the gaps in the ISOs or actually develop new standards for areas that standards are not available. And these RSTs in combination with the publications can actually do that. The next thing that I think is very important for the folks who are on the call today at this conference, that to know that we take feedback on our regulatory science tools. So if you download a tool and you're using it and you find something that you think it's very important for us to know, please, there is a feedback section on that tool. Provide your feedback, because we go in and we check to see how we can improve these tools. So for example, if you ran a method and you got a little bit of a different answer that is concerning, we would like to know that. We also look at the traffic. If a tool is receiving a lot of traffic, that means that we are moving in the right direction. If a tool is not getting a lot of traffic, then that means that maybe we're not doing things right. Maybe we should go back and think why this tool is not receiving the attention it should. So in layman terms, a tool can do a lot of things, and we are hoping to be able to allow for those companies that are maybe younger, maybe they don't have the means of having a huge regulatory group, to use these tools to be able to cut down in the process and help them, with their technology development. That's our goal.
Fantastic, fantastic. Next one, I think probably for you as well, Victoria. Could you comment on an example of a regulatory science tool in the catalog? I know you shared a screenshot and there was a link there, but I was wondering if you could elaborate a little bit more on maybe an example.
So there are a number of examples in our group, the chemical characterization group. Our most recent tool, and I think Billy, you and I have talked about this before, our most recent tool that went on the catalog was May of 2024. It's a tool that has looked at 107 reference standards for materials that goes into medical devices. And we are trying to, our aim is to develop a coverage map for these materials that are used in medical devices. And so this tool is very important because it will tell you, okay, if you are doing a submission, you may want to take a look at this and evaluate your material. The tool is so important that within the first day, the hit on the tool was 4,000 people actually went in and took a look and downloaded. That tells you how important the area is. And we're actually continuing. The next tool that is related to medical device reference standard material is going to come out. We're starting to work on that, and we are going to cover a number of materials. So one would be additives, the next one will be polymers, oligomers, and monomers. The next one will be, colorants. There are, we take all of that into consideration, and a lot of work went into getting the tool together and put it, because we had to look at a lot of chemicals and consider whether these are actually ending up in a device and then manage it from that. There's a whole team that works on that. They're not here today. Omar was partially a part of that team for a while until he moved into the breath work. But yeah, that's an example of a tool that we have recently put on our catalog. 2 more is in the process of coming. As soon as our publications are approved by the journal, the tool package goes into our tool team and they look and review. It gets reviewed, and once we go through the corrections that they ask us to do, and it's approved, and then it goes on our catalog and it's available to the public for free to use it. It's open to public. So that's how it works.
That's great. I think it's interesting because you can see how there's read across in chemical characterization. There are 2 similar types of challenges that exist in the breath space. So it's a wonderful foundation for a lot of the challenges that the breath community face. Next question, maybe for Eric, it kind of relates to some of the challenges within the breath field. You mentioned the point that getting chemical standards, it's expensive, it's hard for small groups to do, etc. I was wondering if you could elaborate a little bit further on what you think some of the historical challenges may have been for breath, and how some of these approaches around confirming the ID, ensuring something is on breath, may help in the future.
Absolutely. Great question. Historically, the challenges were, I'd say, sample collection, capturing all these volatiles without background contamination has and continues to be a huge challenge. Instrumentally, we've come a long way. Now we have high-res instruments, many different kinds to help confirm that identification. But historically, I think it's been sample collection and high-resolution mass spec, so quality of data issues. Now, I think with many different manufacturers making great high-resolution mass spectrometers, and then also GC technology is, expanding with, 2D GC with the ability to separate so many volatiles in a sample. I think that's contributing to our ability to identify many of these trace level analytes in the breath. So I think moving forward, we'll see a lot of confident identification coming out in the literature. I think streamlining the workflow will really help labs communicate and verify identifications so that the whole breath community can work together and move forward and then do these type of quantitative analyses.
Yeah, no, that's great. And definitely you can see, as you say, with the different technologies becoming available, if you know what the compounds are because you have a confirmed ID and the concentration, that does help you use all of these different analytical platforms and then have comparability across data as well, which I think is going to be fantastically exciting for the field. Next question here is probably for Omar. It's kind of 2 related ones. So except Python, are there any other tools such as AI that could be used to predict retention?
Yeah, absolutely. There's plenty of more sort of particular tools used to predict the retention in general. We've explored several different techniques in general when we were sort of in the initial stages of this particular study. So one of the in some of these studies that we've actually have modeled, some of our semi-empirical approaches do take an AI and in essence also sort of like a machine learning type of techniques in general. We're not using all of those very complicated particular type of modeling, because one of the pitfalls that you may fall into is that you have to generate an insurmountable amount of data in order for those particular models, in essence, to sort of work properly. So on our end, that's why we sort of have taken a sort of a semi-empirical approach, and we're not using any sort of complicated, you could say quantum chemistry sort of programming, just because in essence, we're sort of trying to test all these particular methods without generating a high level of, or generating a high level of data. So yeah, there's a lot of techniques out there. One of the ones that are that we also kind of try to model a lot of our things were from neural networks in general. So there's plenty of techniques out there that you can definitely model all of this based out of. It's actually what you use, what works better for your actual type of application.
Yeah, yeah. Next kind of follow on to that, and this is one I was thinking as well. Is there effort to include other mass spec methods other than GCMS, specifically without GC? So I know a lot of the breadth research community will use SIFT-MS or PTR-MS. Do you think there's kind of scope to use these same types of techniques for those other analytical instruments?
I don't know if that's directed at me, but at least
That's at you. I would imagine that one.
So I would not for a particular project that we're doing currently for breath. It's another project that we're doing here internally. We're not just focusing completely on using GC techniques. We're focusing on LC techniques. But as far as what I'm currently doing, I haven't explored any sort of other techniques that sort of don't employ GC. So that's something that definitely I could discuss within my actual work group to see if there's any other sort of techniques we can implement to this type of research. I'm pretty sure there's plenty of other mass spec techniques that we can apply to what we're currently doing.
Fantastic. Next one, I think, is probably for Eric. So you talked about the low res and the high res instrument in the talk, and you outlined our things like GC-GC. Is there a plan to do GC-GC, or will it be using different column chemistries in your analytical work, Eric?
Our short-term goals is to use different column chemistries and to continue our current work looking at different stationary phases. Yes, but GC-GC is not off the table. In the short term, we will have 1D GC, high resolution. But I do foresee, because it's a very popular technique in the breath area, I do see tandem GC in the future. However, I cannot say how soon that will be. But we have an excellent lab here, and we're always expanding our instrumentation. So it's something we've been thinking about.
Excellent. Thank you. And again, I suppose if different groups are IDing and quantitating, then we can coalesce different data sets together as a whole field. The next question is for you as well, Eric. Is NIST library the only reference library being used currently?
Great question. No, we're not just using NIST. We're also using Wiley, Agilent, and other resources out there. We prefer to use publicly available databases to more accurately pull from readily available sources and not favor any commercial databases. So we're looking at as many sources as we possibly can. NIST is the most used, and so we thought that would be the best place to start.
Fantastic. Next one, this is for Omar. Fantastic talks for Omar. Do you think a similar modeling approach could be used for prediction of pre-analytical sampling efficiency, e.g. for prediction of recoveries of on-breath VOCs using different sorbent types?
Oh, that's a fantastic question in general. I was seeing it here in the QA. You know, we really haven't discovered that particular part of a pre-analytical sampling efficiency. That would be also one of those particular topics that we could look more into in general. That's where, and for this particular talks, that's why I take some info in general. So that's one of the topics that we can also sort of dig into for future projects. So as far as in general for different sorbent types, I don't know if the person that asked the question means different sort of columns that we're using for this type of work. If it's more related with the columns, then yes, at least for my ongoing work or future work, I would say within the next couple of weeks, I'm actually going to be trying all of these experiments that we've designed in the laboratory, and we're going to be applying pretty much everything to different types of columns. Analytical columns. So some of the reasons being is that a lot of our modeling also takes into account the packing inside of the column in general, their stationary phase. So we're sort of designing all of these experiments in the laboratory so it could well align as well with a lot of our computational modeling. So hopefully that's what the question was referring to, more like the column packing, hopefully.
Thanks, Omar. Time for two very quick ones. This is for you outside Victoria, and this is something that we've talked about before as well. VOCs, not just in breath, but if you look in the head space of other samples, like a blood sample or urine, you would expect that, you know, that's fundamentally where the VOCs are coming from. Is there plans to look at other sources of VOCs from these other sample types?
Yes, and there are a few things I'm going to cover for plans in the future. Obviously, what Eric and Omar and the team have presented is just the beginning of our work, Billy. I think, and we're focusing predominantly on GCMS because that's where we think that the start has been to develop for breath. You know, the studies are certainly whatever is in literature and what we have found. And if we are going to build the base, you know, you should start where there is some data so that you can take a look. And predominantly, I think GCMS has been something that they have looked, whether it's Orbitrap or something else. Plan for other methods, possibly. You know, I think once we get our handle on this confident identification using GCMS, then if there is a potential for other methods, then we can address that. That would be probably the next level, I would say. I think it's important for us to build the base and then kind of move over. Plan for other areas. Yes, other body fluids are very important because I think it will simply add to the confidence of the breath. If you could correlate, if you could do the work and then correlate the biomarkers that you see in breath with other body fluids, I think that would be tremendous because then it would add to the you know, evidence that one needs for the work. And we have plans to do that. And we are thinking about it. We've just started this work and the group has been moving tremendous. I mean, these guys are every day they're doing something. And we started this in April. And since April, we've come, you know, quite a bit in an area that I have to tell you, none of us were an expert. And just by studying and working and talking and learning, we've brought ourselves to this level. And of course, we are analytical chemists by background, but we had some, you know, steep learning. We are hoping to use TB as a base because it's a disease that possibly it's easier to look at, and there's a lot of literature out there that one can rely on, and hopefully we can cross-check this biomarker to get this confident identification. We're all seeing the same thing. And then we would like, if possible, for us to move to the next and the next and the next. So that's our plan, and that's what we're aiming for, and hoping that we're intending in the future to expand to other body fluids and expand into other diseases as well.
Fantastic, very exciting. And you mentioned earlier that always wanting feedback. I love this last question of the session. It's how can members of the breath community help in this effort?
A lot. They can do a lot. So we're always open to working together if there is a possibility. If there is a situation where someone is using another piece of instrumentation that we don't know about, it would be nice to know about. I think collaborating with the breath community, all of us together can bring that confidence that we need in this area, in this arena. And feedback, however you can add value to this and share, I think it's going to make us better at what we're doing.
Wonderful. And on that note, I want to say a final thank you for all the contributions today. It's been really exciting, and we're really appreciative of you joining us today.
Thank you. Thank you. It's a pleasure. Thank you for having us.